things like immunosuppression.
Sorry. And then we're going to start the webinar in just 1 second. All right, and we'll get started. All right, we'll give people a few seconds to come in and say, welcome everyone to the Renegade Research Clinicians Roundtable. If this is your first time with us, thank you for joining us. I'm Isabel, I'm the CEO of our organization. We are a patient and caregiver-led, decentralized 501c3 nonprofit organization focusing on complex multisystem chronic conditions such as CFS and Long COVID. Within this, we have several areas of focus: research that's agile and patient-centered, education to increase the number of clinicians treating us, and also care and education within that care. Our work is made possible by our wonderful team of volunteers who move the wheels of our organization. Most of us have a personal interest in advancing research and care of these conditions. Today, I want to give a special shout out to Ellie, who has been instrumental in making the Clinicians Roundtables happen. To stay in touch with us and hear about upcoming events, please subscribe to our newsletter and follow us on social media. We will be posting this link throughout the roundtable for you. As a nonprofit, our work also depends on donations of all sizes. You can help us fund our work by donating to our funding campaign and also by sharing our campaign with others who can donate. To raise funds, we also have some affiliate discounts by partnering with companies whose products and services we use and like. This does not constitute advice on using those products and services, and as always, discuss with your healthcare team. But if you do use them, please consider using our discount codes to help fund our nonprofit work. We have some great specials coming up for Black Friday week. We will share those in the comments, but also stay tuned on social media for any additional discounts. Today's roundtable is with the Amatica team. They are on a mission to improve patient outcomes by facilitating personalized care. They are building a new model for understanding and treating ME/CFS, Long COVID, and other complex chronic conditions through patient-centered research, cutting-edge data science with full transparency. And their goal is to ultimately solve these complex conditions. So without further ado, a big welcome to the Amatica team, and you guys can take it away.
Thank you so much, Isabel. Thank you for the lovely introduction, and we're really honored to be here. As you know, we've been fans of Renegade Research since we were just, still trying to figure out the very first work that we did in this area. So it's really always a pleasure to collaborate with you, and we're really honored to be here today. And thank you everybody for joining. So we are going to give an overview into our company, what we do at Amatica, how we approach research, our strategy, and the major focus on today's presentation is to talk about how we is the big picture, how we plan to go from what we do now and the fine detail of what we do to making a real clinical impact. And that is, I think, the kind of central goal. Everything we think about and everything we do is about impacting the clinic. And that can be even modest impact to hopefully something really impactful in the future, significant in the future. So we're going to start our presentation and we'll begin with a little introduction into ourselves and how we met and how we got started. I'm going to start by sharing my slides. And I think everybody should be able to see. Hold on a second. Okay. So yeah, as I mentioned, we're going to give an introduction, a bit about our mission and our strategy, what we actually do, so the types of data that we work with and the tests that we sell, and then how that kind of relates to where we're going with our goals to impact how these diseases are treated and understood. And just to start, we wanted to take an opportunity to introduce ourselves. So, um, on a second. Yeah, so I'm Nick. I'm one of the founders of Amatica, and I've put two pictures up here. So this was me in 2019, leading a workout group by the Kamogawa River in Kyoto. And this is me in 2025 with my beautiful niece, happier than ever. So very gracious to be with her. So before I became a kind of COVID-induced ME/CFS patient, my background was bioengineering and then neuroscience. And so I did a PhD in neuroscience. And after completing my PhD, I worked across cell and genetic engineering projects, mostly in cell therapies and advanced therapeutics like gene therapies. I worked across Japan and several places in Asia. And at the time I got sick, I had just been made chief scientific officer of a stem cell engineering company in Kobe, Japan. Um, and I contracted COVID in May 2021. I had about 5 months of basically feeling like I'd recovered and was quite normal, which I think is an experience many patients have had. I had some symptoms coming and going, and then in September 2021, I did one HIT workout, which essentially triggered an ME/CFS-like disease. Over the next kind of 3 to 6 months, I went down to a place where I could only walk around 50 meters a day, sorry, 50 meters at a time, maximum. And my biggest problem is basically PEM. So I have very sensitive PEM to physical exertion. So I can be like almost quite kind of symptom free at baseline. But if I try to move beyond this kind of barrier, then it can trigger long-term disability, all types of whole body symptoms and fallout. So PEM is really my biggest disease feature. I'm actually very lucky that I don't have a lot of neurological symptoms. So I don't have a lot of brain fog, but I definitely have some neurosymptoms as well. So Jack and I actually met as patients through patient communities. These were communities of patients who were trying to understand and treat themselves. And we started originally just trying to understand our own disease, and that grew into what we do today. And I'm going to let Jack introduce himself now.
So, yeah, hello, I am Jack. You've probably seen me posting on X about all of the Amatica work. So, similar to Nick, this was a photo of me in 2020, where I was quite active before COVID. I participated in a lot of competitive sport, had zero health conditions that I knew about. Now, looking back, I actually had Ehlers-Danlos loss since a young age, but no one had really kind of informed me that that was a risk factor. And then I was originally actually studying aerospace engineering, and I finished my degree. I was heading into industry, and in 2021, I contracted COVID in September. And coincidentally, same month as Nick, I then developed long COVID for me immediately after my infection. Actually, during the latter end of my infection, I went straight into neuro long COVID, developing visual snow syndrome gradually over a few weeks, which is like neurological sort of disturbances in your vision. That then progressed to POTS, a loss of my depth perception, some mild MCAS, fatigue, and some sort of minor muscle weakness. And I then had to move back home to where my parents were in Cyprus. So my life was basically put on halt. First year, I sort of had to come to terms with what is this disease, and especially being in Cyprus, none of the clinicians or physicians had any clue that long COVID actually even existed. So at the start, I was completely unaware of the concept of long COVID. And then in 2022, I sort of came across the name, started researching it myself, sort of started pouring all of my effort into figuring it out. And I read a few reports of positive responsive to immunoabsorption. And then I decided to give that a go. And sadly for me, immunoabsorption ended up being probably the worst mistake I made in my illness. It took me from that sort of moderate where I was still able to kind of go on walks and stuff to very severe for a few months where I was blindfolded and ear defenders for a month straight, no stimulation at all. And then since then, the amount I've recovered, I still have now extreme MCAS. I've been on a diet of only two foods for close to 3 years now. My muscle weakness limits me to about 20 meters of walking, and just a bunch of other neurological problems. But through this process, as Nick mentioned, of trying to kind of then get myself back to a level of functionality, which is still a process now, I met Nick through these disease sort of groups, and we both shared the feeling that this, what we call it, treatment roulette, where a patient has to basically gamble their little remaining functionality on a treatment that has maybe 10 anecdotes or 20 anecdotes online of which you're not even sure if they're reliable. That patients shouldn't be gambling their lives on random treatments, and that there needs to be a personalized approach to this. And we sort of resonated on that idea, and that's where we then progressed to Amatica, which we'll get more into now.
Yeah. Thanks so much, Jack. And I think, yeah, just to add to that, I think Jack's experience of immune absorption, we obviously know many patients who've been through this kind of extremely traumatizing experience of trying a treatment which might have helped someone they know, and then it has a, permanent or really serious negative effect on them. My kind of clinical experience at the beginning of long COVID couldn't have been more different to Jack because as I mentioned, I was kind of doing these high-powered jobs. I was just about to go to this absolute dream job in Japan. So I arrived back in London, and I very quickly connected with the top doctors in the space. Through my networks in London, within a month of falling sick, I was seeing some of the still today most respected doctors in the space. And yet, of course, the experience was still the same. It didn't matter. I had access to these very elite doctors, but at the end of the day, nobody knew how to treat patients at that point. No one knew which treatments would help which patients at all. So again, yeah, that was a kind of really one of our original conclusions. So with Amatica, Jack and I, we started working together on originally this was among our patient communities. We were trying to develop methods to stratify the patients. So even within these informal patient groups, we were trying to come up with ways to separate patients into groups that could eventually predict their response to treatments. And we have been working on this for 2.5 years, and we've been through kind of various iterations to arrive where we are at today. But the starting point is really exactly what Jack just shared, his story and also this wider story that people experience. So, as he said, it's this kind of medical roulette for patients, where you have this population of patients, which is perhaps, possibly the most heterogeneous patient population in the world. I mean, I don't know if there's any disease that has more extreme variation than ME/CFS and long COVID, right? Not just in terms of presentation, we have patients with extremely different phenotypes, but also in clinical outcomes. So, patients who may even seem relatively similar can have extremely different responses to interventions. So, this really heterogeneous patient population leads to two really big challenges. So the first is what we've just said, which is this treatment challenge where, a doctor or a clinic might have many patients who, technically fall under this umbrella of long COVID coming to them. And from a doctor's point of view, from a clinician's point of view, it's extremely challenging because those patients come in and they are sick from since COVID. They know that they're very confident it's post-COVID. We don't have formal diagnostics. So firstly, many clinicians are kind of already very apprehensive to give treatments because they don't have a formal diagnostic pathway. But for the minority of doctors who really do roll up their sleeves and try to treat patients who, I think as patients we couldn't have more respect for, it's extremely difficult for them. They may see 20 different long COVID patients and they could give them all the same drug. And you might see, 18 of those patients have no effect. You might see one patient have a serious positive response, and you might see one patient get permanently worse. So, that this is the problem. So for Amatica, it's this problem. It's the heterogeneity of the patients. It's the difference in outcomes. That is the central problem that we're focused on. the other arm of this problem is in clinical development. So I think, one of the things that probably many patients and doctors appreciate is that, you have clinical trials going ahead all the time in this area. There have been, okay, there aren't nearly enough. There's not nearly enough investment and movement, but there are many historic ME/CFS trials or long COVID trials. And, many of them, or the majority of them, have failed. And that may very well be due to this exact same problem because all these companies that are trying to develop clinical products, who are trying to develop drugs or repurpose drugs, they are once again taking this cohort of extremely different patients and calling them one group, right? So if you think about a standard kind of phase 1, 2A clinical trial, you might have 20 patients advancing into the first treatment arm, right? And so 20 patients, we believe there may be dozens of different subgroups within long COVID or ME/CFS. So 20 patients you may have selected at random from 100 different buckets, and then that is your patient group. So in a classical legacy clinical trial design, you would compare your patient group to your control group. Now, if you just control patients from these two populations like this, firstly, obviously, you're very unlikely to have statistical significance, even if there is a positive effect in some patients, but also you completely miss that information. Like, for example, if you are trying a drug in this patient group, it may be that 10% of patients have a really significant response, but the clinical trial will fail, right? The clinical trial will conclude no statistically significant effect. But for patients, and I think this is really important to understand, if there was a drug that had a 10% efficacy rate, that would be, the drugs of that type of response rate are incredibly important to patients, right? I mean, if many patients will have tried more than 10 drugs themselves. If a drug has that response rate, patients would like to know and access it. So these clinical trials need to be redesigned. There needs to be more sophistication about how we analyze the response of in patients. And that's really what Amatica is focused on, is solving these two arms of this problem. One, how can we help clinicians to match treatments to the patients who are actually going to respond to those treatments? And two, can we help to drive much more sophisticated clinical trial design to actually identify smaller subsets of patients who respond to treatments? So how do we do that? So the way we do that is the first important thing to say is that, we don't believe we believe there is a hard reality under all of this, and biology is extremely complex. I think anybody who's worked in biology knows that. I think every patient understands that just from their exposure, and also within this disease, just how much complexity there is. As someone who's worked in, biology and biotechnology for more than 15 years, I I still get blown away by how much complexity there is in biology. And one of the anecdotes I often share about this is that, even though I'm working on biology every single day, I will invariably come across a system which I have never ever heard about, which is like another layer of depth to understanding how something works. So the point I'm making is that even so within this extremely heterogeneous, this extremely diverse and complex patient cohort, we believe that there will be real biological reasons why one patient responds to one drug and another doesn't. There'll be mechanisms, there'll be physiologies that determine those responses, and those will be measurable. So if we can access that information, or if we can access proxies of that information, we should be able to identify the real reasons, or at least signals that can predict response of those patients. So let me unpack that a little bit. So if we take the heterogeneous patient population, this extremely diverse patient population, we basically apply this a kind of layering on of data approach to try to stratify them into different biological groups. So that works in several stages. So firstly, patients who buy our tests and join our projects, they firstly do, or will do, it's just being rolled out right now, a really extensive a digital questionnaire software platform. So this is more than 350 data points. It's the most detailed disease profiling platform ever developed. It was developed using the patient community. So we originally started posting on Twitter about this in the middle of 2024, and we asked the long COVID community and ME/CFS community to list every symptom they had. So we proposed like some starting point lists, and we said, please, tell us every symptom that isn't there, tell us every detail. And over the course of several iterations of that, we tried to create a platform that could characterize the real complexity of a disease. And what I mean by that is, we don't think it's enough to say I have PEM or I have muscle weakness or I have dysautonomia. We want to know the really specific details of that. I think that everybody in this space probably appreciates that PEM is itself a really complex spectrum of manifestations, right? There could be 20 different types of PEM or even more. Equally with muscle weakness, there are muscle weaknesses with very different phenotypes. Dysautonomia can be extremely complex. So the first thing is we collect that patient profile data, so really deep information on their exact presentation, disease profile, symptoms, etc. And on top of that, their treatment response history, right? So we collect from everybody, or we will collect from everybody, which treatments they have responded to, which they haven't responded to, any that made them worse, etc. So firstly, we get this data, and by the way, just for everybody listening, this will be completely free to use, and we will be honored by every single person who completes this questionnaire, because even that alone will drive a lot of our data science. We believe that even really sophisticated analysis of that level of data, which has never been done before, may already identify clinically significant groups which have, the power to help to improve care. After that initial patient profiling, we then collect layers of different biodata. So we originally started with protein profiling. We're going to go into the detail of this. So that means just profiling the presence, the amount of different proteins in the blood. So these could be signaling proteins, inflammatory proteins. They could be markers of the certain states of different systems like mitochondria or metabolism. But we want to combine this with other types of data. So next, we started rolling out RNA sequencing, which I'm going to explain in a little bit of detail a little bit later. But that looks at every gene expression level in your blood. So that can come from the immune system. It gives very deep functional data. So we have this kind of genetic data telling us about states and functions, the protein effectors of different systems, and those make the foundation. Next, we want to layer on other types of data. So next year, we're going to be rolling out the most extensive autoantibody array ever released directly to patients. It has 21,000 human autoantibody targets. And finally, we will also integrate COVID persistence data. So we're developing a platform of COVID persistence tests that will give us information into, viral load and whether you have COVID persistence. So by collecting different types of data and layering them onto each other and combining that with this really detailed patient profile data, we create this very rich, very highly indexed database that can be used to drive data science. So with this really, really high content data, we are building on top of that data science infrastructure and bringing together data science expertise to drive two main things from that. So the first is deep individual insights into health, and the second is stratification of clinical groups. And these two different things are really important because the way our model works is we want to try to create tests that can give individuals really deep insight into their health that was never accessible before, that can identify real problems in them and can give them answers. And so our goal is that we want our tests and we want these really advanced types of biodata acquisition technologies that we sell as tests. We want them to give people really deep and interesting insights so that people get something very valuable from our tests. And then the second arm is that they know that by buying our tests, they contribute their data to our research to drive this clinical stratification. So anybody who buys our test, in the short term, we want to give them the really detailed insight into their disease, and then that helps to drive this large scale data science, where the goal is that in the next years we'll stratify groups that can be matched to different treatments. So now we're going to talk a little bit about our different tests so far. So we have two tests, and I'm going to hand over to Jack to introduce the 31 marker test. And Jack, I'll go through the slides as you progress.
Awesome. Thank you, Nick. So the 31 marker panel was the first test we released, and it was actually something that I designed over about 1.5 years of doing my own research into ME/CFS Long COVID, past research that had been done, and with a lot of intent in that 1.5 years to look at what I thought might be the key systems involved that you can kind of get sort of actionable insights into. So these are functional systems that we can say this is a tangible thing going on. And it was originally maybe 45 markers, then we scaled it down to hone in on what we thought were the key systems. So this includes stuff such as like mitochondrial function and your energy production, O2 sensing, and that can actually lead back into mitochondrial function. So you have systems such as pseudohypoxia, which is where your body can seem as if it's in a low oxygen state, but it's actually driven by mitochondrial stress. So getting some insight into stuff like that. Vascular function, given there's suggested to be a large element of endothelial and vascular dysfunction in ME/CFS. Inflammatory markers, so we've looked at sort of your interferons here, so interferon alpha, beta gamma, and lambda, and then also there's certain markers that can be initially anti-inflammatory, such as TGF-B1, but when they're chronically sustained in a high level, they can then actually in turn become inflammatory and also immunosuppressive. So within that, also immunosuppressive markers, given the potential for viral persistence within the disease. And then RAS system, which is your renin, aldosterone, and angiotensin system, because COVID's main target is ACU2, which is part of that system. So there's a lot of suggested research that it would be altered, but there hadn't actually been a decent amount of research looking into the actual state of this system. So our thinking was, why don't we, with our method that we can acquire a larger quantity of patients over time, finally sort of shed some light on this system? And then you've got your neuroinjury and neuroinflammatory markers, and autophagy and mitophagy markers, which is the recycling of cells and your mitochondria. There's certain research that suggests in neurological diseases such as Parkinson's and Alzheimer's, there could be dysfunction of recycling of mitochondria. And we suspected that potentially it could possibly be the same in long COVID and ME/CFS. So we thought, let's add those systems in and gain some insights. So that was the thought process of slowly designing this 31 marker panel. And well, from the research, from the results, which we'll get into later, we managed to get a lot of significant and novel findings that have been yet to find. Do you want to go to the next slide, Nick? So we've now run our second batch, but from our first batch, we finally put our process together of taking the patients, running them against control, and then doing our deep stratification. So we use various methods where we would take the patient's raw markers and use mathematical formula to determine the similarity between patients' marker profiles in an unbiased way. So we're not selecting patients and saying we think that you've been a cluster. We take their markers, and For example, in this image to the right, we took what we thought would be neuroinflammatory markers and neuroimmune markers involved in that system. And we did clustering of patients using those markers. And what we found was really interesting. We found three subgroups of patients which stratified as one group having higher neuroinflammatory markers than the rest of the patients, another group having more mitochondrial immune markers, and then a third group that didn't actually seem to have much presentation going on, if anything, a reduction in inflammation. And this just goes to show the sort of variation that exists within this patient population. If we were to take all of these patients together and cluster them together, it would mean we would not get these deep insights. And it could be that the patients with the neuroinflammatory presentation would be your patients that you would want to enter a clinical trial or respond to a drug in a clinical trial that's targeted at brain inflammation. And then likewise, your mitochondrial patient subgroup could be those that would benefit from mitochondrial support. But clustering all of those patients together, as mentioned before, you would not get that statistical significance because the clusters are quite small for statistical significance on their own. So this was when we got our first sort of proof of concept of the success of the model. It was not a huge amount of patients, but since we've had batch 2, we've managed to scale it to larger, and we'll get into some of those findings a bit later on.
Yeah, thanks, Jack. Hold on a second. Just lost my mouse for a second. Yeah, so so we spent the last year or so developing and rolling out our 31 marker tests. I think one point Jack makes that I think is really important is that, we are really data driven, so we don't have any kind of preconceptions about what we think the disease should be like or will fit into. We obviously have lots that we think about, but that's really important to us. So the second type of test that we rolled out is RNA sequencing. And our RNA sequencing, we partnered with University College London, which is my old university where I did my PhD. And this is one of the most powerful techniques that's used across biotechnology industry, but hasn't really been used in any patient-facing capacity yet. So the way it works is in our case, we collect whole blood samples. RNA is purified from them, and I'm going to give a quick crash course in what RNA is and why it's so interesting. But we take whole blood, we collect the RNA from that whole blood, and then that RNA is sequenced, and the data we get is basically we can find out the number of copies of every gene that has been made. So for every gene that you have, we get this copy number. So why is that interesting? So quick crash course and full disclosure that I am a 15-year plus RNA nerd, and this is my favorite topic, but I really hope I'll make it understandable. So RNA is really interesting. So DNA, I think most people maybe understand what DNA is. DNA are these instructions, this code that carries the instructions to make proteins. And proteins are, the molecules that make up most of your body. We are just really protein with minerals. So every single component, every molecule of your body is generally a protein and has a DNA instruction code in the nucleus of your cells. So that code, the way it's used to make proteins is copies of that code are made as mRNA. So these messenger RNAs. So an RNA copy is made of a DNA gene. That RNA is then taken outside of the nucleus to cellular machinery called ribosomes that basically move along that RNA copy of a gene and match the codons, match the sequence to amino acids. And so those amino acids join together, and then the amino acids fold into the final form of protein, which has its final function. So the reason that analyzing RNA is so interesting and carries so much information is because, if you do a genetic test, you can find out which genes you have, but which genes you have is much less interesting than knowing what genes your body is using. And so when we do RNA sequencing and we find out the copy number of each gene, that tells us how much of that gene your body is using. And so I'll just give you a very simple example of why that's interesting. So if let's say you take a gene for an inflammatory cytokine, so this could be TNF alpha or some really strong inflammatory molecule in your body. If you are in an unstimulated, non-inflammatory state, then there will not be very many copies of that gene because your cells are not producing so much of that gene. Whereas during an inflammatory state, your cells will turn on that gene, if you like, and start to produce many copies. So finding out the copy number of each gene gives actually very rich insight into how our bodies are working. These genes could relate to mitochondrial health or cellular stress responses or immune states. So the by analyzing RNA, we get this very powerful insight into function. And then there's some advantages of RNA, which is that um, it's it's quite reliable information. The copy number is quite is something that you can measure quite accurately from every gene. So it means that when we take a blood sample and sequence it, we get 20,000 plus gene expression levels. So we get, basically your gene expression level for every gene that you have. That maps onto more than 1,000 different pathways, different biological pathways, and that can give really powerful insight. So just to give like some examples to try to kind of give insight into how interesting this can be, um, if you think about, so, we think that mitochondria may be dysregulated, there may be mitochondrial problems in ME/CFS and long COVID. Now, with protein analysis, you can maybe look at some mitochondrial proteins in the blood and try and get some insight. Maybe globally, the best mitochondria test you could get would be like mitochondrial respiration. It tells you how much energy your mitochondria is using. With RNA sequencing, we can pull out, 100 or 200 genes that are involved in mitochondria, and we can separate them into subsystems where we can say, okay, these are the genes involved in mitogenesis, the creation of new mitochondria. These are the genes involved in mitochondrial trafficking to movement around the cell, or involved in the energy production process inside mitochondria, or involved in its shape or its, ability to function. So we're able to separate out individual systems from something like mitochondria and then gain insight into how those systems are functioning. So that's just one example, but you can apply that same logic to basically any system you have access to. So we could look at every gene involved in regulating different immune states. We can look at genes that are specific to different types of immune cells. We can look at the states of signaling pathways, stress pathways, and then just something really interesting that I couldn't help sharing because it's one of my favorite things that I researched this year. So when we're doing this analysis, as I said, we're collecting whole blood. And so from that whole blood, we extract all the RNA, all the copies of these genes that tell us how many of each protein are being made. That information is from the blood, right? So it means mostly we're collecting immune system genes. Now that's great. That's super interesting for ME/CFS and long COVID. But what's also cool is that there can be spillover from tissues. Okay, so there could be endothelial genes which are spilling out from endothelial cells in the side of your vasculature. There could be gut genes that are spilling into your blood as well. Or from the CNS, the brain, central nervous system, there could be genes spilling out. And this often happens in exosomes, which are these little lipid particles that your cells can release. And there was a study from last year where they collected these extracellular vesicles or exosomes, these little lipid particles. They purified them from the blood and then they sequenced them. So they purified these vesicles, then sequenced the RNA inside them, and they were able to detect brain-derived mRNAs. What does that mean? It means that in the blood, they could detect genes that are only expressed inside the brain really accurately. And by looking at those genes, they were able to diagnose they were able to see a clear difference between mothers who had postpartum depression versus mothers who didn't. So in this data in green you have mothers who didn't have postpartum depression, and in red you have mothers who did have postpartum depression, and you can see that they were able to detect a really, really strong signature. The reason I highlight this, and I hope I do a good job explaining it, but the reason I highlight it is because it's really exciting that by collecting whole blood, we may be able to see gene expressions from within the brain. So when we saw this study, we currently have some pilot RNA sequencing data of 17 patients. And so we quickly got up our data and we were able to detect these brain-derived genes in our data set. So it means that this RNA data that we have coming, we may be able to see directly into neurological states via this kind of genetic spillover from the brain. And that same logic we think will be able to be applied to, like I said, bone marrow, endothelium, gut, muscles, etc. Um, so our first RNA sequencing cohort, and if there's any patients who have joined it with us today, firstly, thank you so much for joining. We are sending our first major cohort of 100 patients and 60 age-matched controls to our partner UCL Genomics, who will sequence them, and we're expecting the results back in January. That will be, for us as a company, that will be a really important moment because having a data set of over 100 patients, I think many people listening will appreciate that in ME/CFS, that is a really significantly large data set. So we believe that we may make many findings from that. And then when you think that we have all of this really deep questionnaire data to layer on top, we're optimistic that we may find signals that can separate out different types of the disease or different treatment responses. And so on that note, we are shortly going to talk through some of our most interesting findings, but I thought I would just also share what we're developing. So this is the our pipeline of different types of testing that we're developing. And so as we've covered so far, we have our 31 marker panel, we have our RNA sequencing. Next up, we're rolling out a COVID persistence test. We think this is really important. We think that knowing, the amount of COVID in someone may be really important information. We've been developing this test to be able to detect a single copy resolution, so it will be able to tell you viral load, and also really importantly, we've developed this test to be able to detect which strain of COVID any persistent COVID is from. So it could tell Alpha from Delta, from Omicron, also whether it's from COVID or the vaccine, and the replication state, whether it's actively replicating. So we think that should be really interesting information. In the early part of next year, we've nearly finished developing, and we had volunteers from around the world with really high signals of persistence who sent us samples, and we're running those right now. Early next year, we'll start to release some of our RNA sequencing results and these different RNA sequencing panels that look into neuro states or mitochondria, etc. In the first part of next year, we're going to roll out the most in-depth autoantibody array that's ever been available to patients. This is a method where some incredibly talented scientists developed a piece of technology called HuProt, Human Protein Microarray, which has 21,000 human proteins in a kind of microarray chip, and you can put sample patient serum onto these chips, and then you can see if that patient has autoantibodies against 21,000 different human target proteins. So again, as super bio nerds, we are incredibly excited to see this data, but we think it's going to be really interesting because the a potential split between a kind of autoimmune versus a persistence versus maybe a kind of dysautonomia types of this disease may be really important distinctions. So by getting this really rich autoantibody data, that may give us really interesting insight. And the people who developed this have worked with elite researchers all around the world, including, for example, Iwasaki in long COVID research. And one of the things that they told us as we've been planning our collaboration with them is that in multiple sclerosis, they have found, for example, that there are autoantibodies derived, EBV derived, or EBV uh, mimic, I forget, molecular mimicry. So EBV molecular mimicry autoantibodies in 25% of patients. So that's a super interesting finding. You know, it suggests that EBV reactivation may have a relation to MS pathogenicity, to how a disease starts. And if we start to collect this data and we can pass into hundreds of samples, you know, we may find similar findings in long COVID and confirm other people's work in this area, like Mio Iwasaki and Carmen Scheibenberg and etc. Later, we plan to roll out more sophisticated proteomics. So this will be looking at thousands of different analytes. This could be maybe in the second half of next year. So that will be layered on top. And then finally, taking inspiration from that study I mentioned that was looking at postpartum depression, we will probably purify these lipid particles, extracellular vesicles or exosomes from the blood and try and use them as a way to kind of as a window into tissues from the blood. So really interesting things that we really look forward to sharing with you all. So Jack's going to take over to talk through some of our most interesting 31 marker results.
Awesome. Yeah, thank you, Nick. So from our results, these are our batch 2 results. So the latest ones we got, it ended up being, I think it was roughly 62 patients and 18 control. And it's 32 markers actually, so 31, but TGFB, we run both as total and free TGFB1. And we took some base questionnaire data from these patients. We haven't done the full questionnaire data on them yet, but that allowed us to get some rough sort of PEM data, some symptom data, and we'll get into all of that now. And something else that's very important to us, patients in our disease do not feel the same day to day. And one of the main reasons for this is PEM. So a patient who is taking a blood draw in PEM will not have the same state when they are out of PEM. So we also made it a priority to sort of note the PEM status of patients during the blood draw. So this is a sample from a patient in PEM. This is a sample from a patient who was not in PEM, the severity, and we'll get into some of those results now. So from the 31 markers, we found as a disease total, 3 significant results. Now this was ROC1, angiotensin II, and then free TGFB1. So just sort of a bit of these. So the ROC1 is quite interesting. It's a pathway involved in many things. So it's like immune function, vascular function, sort of structural stuff. And I was initially actually expecting it to be high in long COVID and ME/CFS, but the data sort of does what it wants at the end of the day, and you have to take it on and sort of understand what that means. And interestingly, we found reduced ROC1 in patients and quite significantly. Now, whether this means reduced activity of ROC1 is still sort of an unknown question. So there was a paper in acute COVID that found in extracellular vesicles, which Nick was just discussing a moment ago, that they also found reduced ROC1 protein, but this was actually due to this thing called cleaving by caspase 3, where basically the ROC1 is cleaved and then it does not get picked up by the ELISA, which is the testing method we use. And this cleaved version of ROC1 is actually a more active version of ROC1, a continuously active version. So in this case, in that study for acute COVID, reduced ROC1 protein actually meant increased ROC1 activity. So we will have to do sort of further research to kind of understand, is there a correlation between caspase-3 and ROC1 in our data? Can we look in the RNA for upstream and downstream pathways from ROC1 to see whether there's activity there? But that's quite interesting. And then the second marker, which is angiotensin II. So this is part of your RAS system. So I mentioned before that COVID's main target is ACE2. And in your RAS system, sort of a basic sort of rundown of how it works is you have renin. Your renin goes into Ang-I. Your Ang-I is converted into Ang-II by ACE, and Ang-II is converted into Ang-1 to 7 by ACE2. If ACE2 function is not working, you would expect to see an increase in Ang-II, which is sort of the suspected state that you'd see in long COVID. We saw this increase in Ang-II in our patients, but what we didn't see was a reduction in Ang-1 to 7, which is what you would expect if ACE2 wasn't working. And in fact, we actually saw also a trending reduction in ACE. So basically all of this together potentially indicates that we could have a ACE independent Ang-II increase, which can be from pathways such as mast cells, and endothelial sort of spillage of chymase. Again, more research is needed there to kind of look into that further, but it's important to look what is causing this to kind of understand the upstream mechanisms. So taking the whole system together allows us to see, is it a classical presentation or is it an atypical presentation? And it was a little bit of an atypical presentation, which I guess matches the general presentation of long COVID as a whole. So the following one is free TGFB1. So interestingly, total TGFB1 is where it just measures the total protein count of TGFB1. And that total amount of TGFB1 is not actually active. So it exists, but it does not interact with your body. That was found to be normal in patients, but specifically what we tested as well was free TGFB1. Now, free TGFB1 is the active form of TGFB1. So this is the version of TGFB1 that is now interacting with your body. And that we found a significant increase in free TGFB1 in the general patient population. It seems like it was maybe a sort of subgroup of patients who happened to have it raised, whereas there was still a decent portion who have overlap with the control. And TGFB1 is one of the more interesting sort of proteins involved in ME/CFS and long COVID. It's been consistently found raised in other research and then not found raised in certain studies. So potentially the difference between free and total TGFB1 could be one of the reasons that this is happening. But its involvement is largely sort of like immune suppression, potential mitochondrial function, in some cases neuroinflammation. And what's most interesting about TGFB1 is initially when it first arrives in your body and it's first increased in your body is it's actually protective and it's anti-inflammatory. But when it remains in a chronic state raised, it starts to cause consistent ongoing problems. So it can cause like NK cell exhaustion, which is where the immune system, the immune sort of markers in your body, they stop being able to clear viruses. And you also see it in cancer where you stop being able to combat tumors. So it could possibly be a protein that's involved in such a stuff like viral persistence. Again, you would need more research to see if there's a correlation between viral load and that subgroup, but it's an interesting avenue to look further into. Now, I also mentioned that we collect PEM status of patients on blood draw, and this was one of the more interesting findings. So ROC2, which is similar to what ROC1 in function, a bit more vascular and a bit more neurological. We actually found that patients who drawn their blood during PEM had significantly reduced ROC2 compared to patients that drawn their blood when they were not in PEM. Now, whether this has a similar presentation to similar possibility to ROC1 where it could be cleaved. There's not as much research, so I can't comment, but it's definitely an interesting avenue to look further into and see if ROC2 eventually, again, with the RNA data, whether we can look further into those systems, see if the RNA is lower or higher, what is downstream, so we can get some more information there. But that was a good PEM finding that we actually got in our data. Now, do you want to go to the next slide, Nick? Yeah, so this is the ROC2 data reduced. And then ROC1 as well, interestingly, had a trend to reduce. Now, it's not as significant. It's not significant, actually, and it could just be noise. But it is possible that maybe the row sort of system entirely is experiencing a reduction in sort of values during PEM. And if we go to the next slide, we can actually talk about an individual patient. So we had a patient who generously submitted their blood both in PEM and out of PEM. So their initial sample they sent us was when they were in PEM, and then they sent us a second sample some weeks after when they were now out of PEM and rested and made sure they were okay for the blood draw. And then we ran both of these samples together in batch 2 under the same settings. They were shipped under the same process as well. So we then compared the markers, and what we saw most interestingly was free TGFB1. At baseline, when they were not at PEM, they had virtually no free TGFB1, but then at PEM, they actually went to having one of the highest levels of free TGFB1, potentially indicating that maybe the total version turns into the active version during PEM state. Again, we would need more PEM patients to confirm whether that's actually the case. We also saw a reduction in BH2 and BH4 during PEM. And then the graphs are not on this, but interestingly, in the RAS system, we saw from no PEM to PEM, we saw a reduction in ACE and a slight increase in angi, which further potentially sort of indicates that non-ACE dependent angi sort of generation, which is an atypical presentation. Interferons, most interestingly, you might expect them to go up. What we actually saw was every single one of their interferons had a slight reduction in PEM. And then mitose stress markers had a slight increase. And then again, sort of contradictory to what you might expect, the neuroinflammatory and blood-brain barrier markers, they actually saw a reduction in PEM. Now, these could just be day-to-day variations for some of these markers. We can't comment for sure until we have more patients, but interesting nonetheless. Now, we also took some, I mentioned we took some basic sort of disease profile data from our patients, and then this comes into our deeper stratification. We provided sort of classifications for severity, so mild, moderate, severe, very severe, and gave sort of functional guidelines to those. So a very severe patient would be bedbound continuously. Severe is mostly bedbound with a little bit of house function. Moderate is housebound with a level of ability to leave, and then mild is sort of you can live your life to a level extent, but not fully. And this also comes into one of the great things we have actually managed to achieve. We managed to get a lot of severe patients into our testing through sort of mobile phlebotomy and picking up their bloods direct from their house with medical couriers. So that was a huge thing because I myself am actually more or less entirely housebound. So it was a huge thing that we wanted to focus on, getting these severe patients into research. And then what we actually saw in our severity data was that tweak, which is a inflammatory marker, but has been found raised in some MS patients and has been associated with exercise intolerance, which is not quite the same as PEM. But interestingly, the patients with it raised, there was a subgroup of patients with raised tweak against control. They were all in the severe and very severe group. And then for ROC1, what we saw was there were some patients with levels not near the zeros and the low levels. They were mostly our mild patients, and then as it went more severe, it gradually sort of like decreased. So possibly ROC1 could have a continuous correlation with severity, or maybe it's a sort of a checkpoint in the disease when you go from your mild to your moderate onwards. It's a point check and stays that way. And similar for tweak, it could be that once you get to a level severity, it then starts a new function, or it could be a gradual increase. We need more data to know for sure. So something else we've done, we do with our data, which is part of the deeper analytics we do, is we use Euclidean distance modeling, which is basically where you take the markers, you select them for the systems we want. So this might be like your mitochondrial function, or you take your immunosuppressive markers, or just your RAS system markers, and then you use Euclidean distancing, which will take these markers from the patients and cluster them by how close they are to the other patients. So if a patient has virtually the same raw data as another patient, they will be extremely close together. If a patient has 5 out of 6 of them really close, they will also still be close together. And then those who are quite far will be on the opposite side of the clustering. And in doing this, with our clustering, we then compared that to disease presentation. And what we found was that when we clustered the neuroimmune markers, so this was like NEFL, your S100B, TGFB1, and GI, which have sort of prior research linking them to neuroinflammation, we found that it separated into 5 groups. And from those groups, cluster 1, so group 1, had quite minimal tinnitus. So it was actually 60% of the no tinnitus patients, well, roughly 60% of the no tinnitus patients were all in group 1, whereas only 20% roughly of the tinnitus patients were in group 1. And then group 2 to 5 onwards had a much higher level of tinnitus patients, potentially indicating that in group 2, 3, 4, 5, there is a presentation of markers that relates to tinnitus that group 1 does not present. So we can look further now. This was what we'll be doing in the coming weeks, in the coming months, is we'll be looking further into these clusters to determine what might be the marker presentation that is causing this separation in tinnitus. We also separated marker patients by fibrosis, which is so fibrosis is kind of scarring in the body, so it's built up of scarring. And sort of markers that are involved in this are your typical, they start off as protective, and then if they're left around for too long, they can often end up causing fibrosis, which would be TGFB1, TGFB2, TGFB3, activin B, PGE2. And what we found was, when we compared this against PEM status, that in group 1 and 2, they had not much PEM presentation. So the majority of the non-PEM patients were in groups 1 and 2. And then in groups 3 and 4, there was a much more consistent presentation of having being in PEM at the time of the blood draw. So this could potentially indicate that during PEM, there's an increase in sort of fibrosis markers, but we have to look further into the data to see what exactly is causing that presentation, because it might be an increase or decrease in markers, which we'll look into further. And then, so this is two patients now. One of them is me, which is on the left, and then on the right there is patient A, which is a friend of mine I've met through the patient community, now known them for over 2 years, and they are probably the single patient who I have found who has the closest presentation of disease as myself. We both share a very specific type of narcolepsy. We both have had, thankfully theirs has gone away since, but we both had muscle weakness, which is very specific to a lactic acid type muscle weakness, similar to when you do too many reps at a gym, and then you start to get that feeling even from lifting a light weight. We both have MCAS, and treatment-wise, this is the most interesting. We both worsened from immunoabsorption, and we both had a negative response to Valtrex, which is a herpes antiviral. As you can see, they are actually more severe than me in general disease, but they've had the disease for a lot longer than I have, into the decades. As you can see from our profiles, we actually have the same systems above control. Theirs is just more amplified than mine on everything other than interferons. So this could possibly indicate sort of a cluster beginning between myself and them being more so on the severe side of that cluster. But considering we went into this with the idea, I wonder if we might end up having a similar profile, it was very interesting to see that we actually had the closest profile to each other from anyone else in the initial cohort.
Yeah, thanks, Jack. And I think just one thing on this. So the way this was put together is, so as Jack mentioned, so I think one of the things that we really pride ourselves on doing is we try to analyze data as deeply as we can. And with the 31 marker results, as Jack mentioned, we split out from them subsets of those markers that relate to different systems. And then we did clustering within those to separate patients and to look for more or less similar groups of patients. What you're seeing here is a single radar plot from all of the clustering. So each node here, each axis here is an individual subset of the 31 markers which has been clustered or we've calculated the average score, there's kind of overall score for each patient for each set of those markers. So when we compare these two together, it's basically saying that the overall combination of the markers to do with energy, metabolism, and oxygen sensing were either this different or different in each direction from the control, which is the green dotted line. So really interesting, as Jack said, that we saw such a reflection of profiles across so many different markers and so many different systems from two patients who had such a big presentation. And I think a big theme of today's talk is that we're presenting data which is only really starting to have meaningful statistical power, and we still have a long way to go, but it's things like this that I think give us some early insight into the validity of our methods when we've applied basically mathematical methods to try to extract meaning from these markers, and it relates closely to something like this. So on that note, and with an extra layer of caution, because this is really preliminary data, but we also have been looking at treatment associations. So this will, in the next years, this will become the biggest focus of our work, right? So I think the majority of the work we do in the next few years will be looking for signatures, marker profiles, disease profiles that relate to treatment responses, to matching drugs to different patients, or improving clinical trial design. And so right now we're looking at the very earliest data, and I want to make two really important disclaimers about this. Firstly, it's obviously it's this is not designed like a clinical validation, right? So this is looking at the results that we have from the patients and then their historical report of treatment response. And also this is not predictive, right? So when you do clinical validation of a diagnostic, which is what we'll do eventually, when you do clinical validation, you take the prediction from you identify what you think can predict treatment response, and then you apply it to a new set of patients, right? So in that way, in the new set of patients, you can then determine how good was it at predicting, at diagnosing that disease, false negative, false positive, etc. So this is not a predictive diagnostic at all. It's a very long way from that, but we were able to find some signatures that correlated with treatment response, and they were really interesting. So we started with just the two treatments that were the most tried by patients because as Jack mentioned, we have 62 patients in this cohort, but I think we have complete uh, data sets from maybe 45 of them, 50 of them. And then within that, say 50, there's again a subset of those who have tried LDN, right? So maybe 60% of those have tried LDN. So here, maybe we're looking at 20 to 30 patients total. And but it's still interesting, right? And so what we see when we look at this is we took our data sets and we separated the patients into people who either had responded to LDN or who hadn't responded. So this was all people who tried LDN, low dose naltrexone. Sorry, I assume everybody knows what LDN stands for. So by splitting patients like this, we're able to see that there were differences in some markers between LDN responders and non-responders. And I think the most interesting was that we found significantly lower circulating serotonin. So we had significantly less TGF beta 2, kind of maybe trending less TGF beta 1, but quite significantly less serotonin. Again, the statistical significance is relatively strong, but this is still very preliminary. We expect that by this time next year, we'll have hundreds of patients that will be able to look at these effects in, but still really interesting that we identified different features between different patients. We also applied the same clustering methods to patients to look for signatures that are associated with treatment response. And so, for example, here, this was clustering done by a group of markers that we kind of refer to as injury repair checkpoints. So this was tweak TGF beta 1, free TGF beta 1, activin beta, follistatin, PGE2, and alpha negalase. And so when we cluster patients by those markers, they separate out into these different groups. These graphs, they basically map a higher dimensional space into 2 dimensions, allowing us to visualize difference. So the closer something is, or the further apart something is, tells you how similar or different their marker profiles were for these markers. And for example, by separating patients by these markers, there is some degree of separation of LDN response. And again, this is really preliminary and it's not predictive at all, but we do see groups that have higher response rates. So for example, group 1 here, which I think is this one, has of people who tried LDN, 77.8% were responders. Group 2, which I think is this one, was 75% responders versus group 3, which was 28.6% responders. Now, this is not very sophisticated data yet. And, as I said, this is not predictive. It's not a validated test. And even if we took it at face value, these are not super high precision numbers, right? We're not talking about 99% specificity. Now, we hope to be there in a year to be able to predict treatments with 99% confidence. But I think the point I want to make is that in ME/CFS, even this would be a huge even this would have a massive impact on patients, right? So if we can, for several different drugs, separate people into groups, or we can say that they have a 80% chance of responding. And again, that's not what we're saying here, but if we can do that, that would be huge for patients. So even though this is so preliminary, it is still really interesting and it does give us an insight into where we're going. And I think the other point to make is that it's not just about predicting responders, it's also about predicting who will get worse. In fact, that's probably more important, right? So, our goal will be that when we have hundreds of samples, because again, this is really only a subset of patients. So this is 28 patients, I think, who've tried LDN. So when we have hundreds of samples, the goal would be that we'd say that we'd be able to split patients into groups and say, okay, you belong to a group where in that group, 80% of people respond to LDN and only 1% have a negative response and, or only 0.5%. That's the type of goal that we have as a company because if we can give clinicians those type of tools, that type of information, that could save, potentially huge numbers of patients from negative outcomes or match them to treatments that could improve their quality of life. So, if you think that you're going from a position of complete treatment roulette where you have no idea what's going to happen to then saying, okay, we have a 90% confidence that you're going to respond and only a 1% or 0.5% risk that you'll have a negative response. That will, if we can achieve that for 5 or 10 or 20 drugs in the next 2 years, that could have a major impact on millions of patients' lives. So I hope that gives some insight into exactly what we're trying to do as well. So looking at the second treatment we found some statistically significant differences for was antihistamine response. So again, the reason we're looking at only LDN and antihistamine response today is because these were the most tried treatments, meaning that for our, still relatively small data set, we had enough to be able to make at least some slight, some comparisons. And for antihistamine response, we saw that responders on the right had trending higher ROC1, had significantly lower TGF beta 2, which is interesting, and also had kind of trending lower BH2. So again, these are preliminary, and we now already have enough patients signed up that we know that in the first part of next year, we will have a lot higher confidence on these. So we look forward to sharing those results as they come in. So those are the key results we wanted to share today. And yeah, as I said, keep watching this space because we will have a lot more interesting results coming out in the next few months. Lastly, before we open to questions, I wanted to just uh, go over our clinical strategy and how we plan to take our research forward to making an impact in the clinic, and especially because for the clinicians who have joined today, one of the key things that we want to say and we say to clinicians all the time is that partnerships with good clinicians, with good doctors, is absolutely key to us, right? And so, if we go back to what we started on, this kind of process of very different patients going to a healthcare provider and trying a drug, but then having very different outcomes, as I said, and as we've been trying to explain, our goal is to change this, but the information, the outcome data from those patients is absolutely critical. And so right now, what we want to make as many partnerships as possible with the doctors, with the clinicians who are treating patients in this space. And we have, relationships with some of the best doctors in the world who are taking responsibility and working with patients, making them fully informed, but using the tools that they have to try to treat them. And those clinicians who are prescribing treatments off label, we want to partner with them because if they will be treating patients anyway, trying the best drugs they have available, then for us, we need to get in and get the data that goes with that clinical process. So that means collecting the patient baseline, their questionnaire disease profile, and then their biodata. And then after during and after they've tried the treatment, monitoring their outcomes. So, you know, I guess there's kind of two ways that we work, which is one, every patient who contributes data and that we hope we give, really meaningful and interesting insights, their historic reports of their treatment responses is really valuable, right? That immediately gives us some information to go on. But it's historical. When we work with clinicians, and we have, I think, probably, 5+ close relationships with clinicians like this already. But when we work with clinicians who are actively treating patients, that data is much more powerful because it's now. It's not historic. It's not what happened in the past. It's what is helping patients now at the point they are today. And it is longitudinal by nature, right? We have a starting point, we see the outcome, and we collect the data. So, it's kind of, I think, it's like we try to, this clinical space where clinicians are trying different things. uh, it's critical that we collect data that can mean that the responsibility and the effort made by those clinicians and the risk made by the patients to try different treatments to try to improve their lives, that can contribute to everybody improving together. I think someone said it very beautifully recently, or some people have said, all the boats rise together, right? So each of us who tries different treatments, if we contribute our data, whether that's just for questionnaire data or for those who can to do our protein or RNA tests or the other tests that are coming, by contributing that data, we pay it forward to help future patients have a better chance of responding better. Um, so these clinical partnerships are central to that. And we encourage all clinicians to get in touch with us. And if patients, if you have doctors who you think are doing good work in this space or helping patients in any way, we really appreciate connecting. And our goal is basically to create the tools for those clinicians that allow them to predict treatment response. So the change would be that instead of the patients arriving at the clinic and then the outcomes coming after treatment, we want to be able to split patients into those groups before they see the doctor, and then the doctor has in front of them the data from us where we can say, okay, this patient has this probability of responding to this drug, this probability of a negative outcome from this drug, and then that can improve outcomes. So I hope that everybody has found this interesting today, and we really appreciate the invitation here from Renegade Research, our friends there, and we really look forward to any questions and discussion. Thank you.
Thank you, guys. This has been so rich, so much to go off of here. We have a lot of questions, and since we have a limited time, I'm going to go ahead and say, if we don't get to your questions, please email me, Isabel at renegade-research.org, with your question, and we'll do our best to get it to the Amatica team and post the responses on the recording. So just want to disclaim that since we have so many questions. We're going to start with Dr. Hutton, our Chief Medical Officer, with a question and comment.
Hi, yeah, just amplifying wonderful presentation and sharing the details of how it all works. I have a couple questions. One logistic, about how long does it take to complete the 300 question survey? And if somebody goes in to do it, can they do it at more than one session, or is it all at once? And
Yeah,
then I have a couple quick questions about some of the clusters.
absolutely. So yeah, great question, John. So from the beginning, we've tried to design this to be as accessible as possible, right? And we're very aware that some patients may not be able to handle it, but what we've done is, so it's a software platform. It saves every keystroke you make. So you could be halfway through a question, you could throw it down and go to sleep and rest and come back and it'll be right there. So, my kind of take on this is, even if it takes you weeks, it doesn't matter. If you can get it in, it's super valuable and we appreciate it. And then also for patients who that's still too severe for, we're creating a shortened version as well, which is just the highest level things as well.
Great. And then on the clusters, I wondered if the information from your survey and history would let you know if, for example, cluster two, which was non-inflammatory, and cluster three, which was inflammatory, whether people went through an inflammatory path to then get to a non-inflamed path, for maybe hypothesizing that fibrosis might be taking over after inflammation.
It's a great question, and I think we very much see it like that. It's one of the things about this snapshot of people's disease where we see it, we don't know the history. But Jack, maybe you want to comment a little bit on this. It's something that I know you've thought about a lot.
Yeah. So right now we don't have all of the data in for those sort of subgroups, but one of I think probably the best sort of ways to kind of gain some insight into this will be the duration of a patient's disease,
Mhm.
and eventually looking at the correlations between the duration of their disease. We haven't got that data immediately right now, but it will be in the next few weeks, months that we will have it. And then we will continue to do the analysis on these patients as we move forward and as the database expands. So hopefully some more information in the near future, basically.
That's great. It sounds like you're collecting a lot of this information in terms of raw data that can be analyzed later on and tied together.
That's
Um,
exactly it.
and then the last question is on the TGF1, or looking at free TGBF1, it looked like there was a distinct group of three controls that were much higher than the rest of the controls. And I wondered if your controls are going through those surveys as well, and if those might have been occult people with disease. Yeah, that one, the third one over from the, yeah.
Yeah.
Yeah, so on this, we actually have the belief that a healthy control isn't really a thing. You just have a control, and that person probably has a multitude of issues that they may have themselves. So we will be asking control to go through the questionnaires as well, for sure. We actually in our I can actually comment on this in our first group. We actually had two of the control coincidentally who then ended up in the months after drawing blood, ended up having moles that were found to be cancerous. So whether that affected their blood draw, no way to know. But it just shows that it's definitely worth collecting that data, and you can never be sure.
Thank you very much. And I know there are a lot of other questions too, but just fantastic discussion today.
Thank you so much. I just wanted to follow up on what Jack said. Yeah, just to add to that, we really don't believe in the concept of healthy control, and like one of the ways we think that when we collect healthy control because it's really important clinically, right, for clinical development, and it's something that we all understand. But actually, when we analyze, we're much more interested in separating phenotypes, right? So we want to say, okay, because definitions can become a flaw in a lot of experimental design, right? So we think, okay, instead of saying patient versus control, let's say people with brain fog versus people without, and we think that may actually hold a lot more information than just the binary comparisons.
Okay, thank you guys. All right, so let's get to the full list of questions. First question is, how can it be ensured, especially with regard to viral persistence, whether an acute infection was present at the time of the blood collection? That should actually be relevant, correct?
It's a great question. I think so. There are many pieces of information which we would like to have, and I think when you do research like this, you're constantly fighting to try to have confidence in your information. There could be confounding factors during blood draw, and things like an infection absolutely could be there. I think that when we really reach the kind of integration of the different types of data that we plan to, one of the advantages we might have is that we should be able to see signatures of these things. So, for example, when we have 20,000 gene expressions, if you've done RNA sequencing, that there are pretty clear signatures of an active infection. So, let's say we grow to 1,000 samples, and then on our collection forms, we actually, in our data collection, we actually have, 50 people say that they had an active infection during a blood draw. We will be able to see whether we can see a signature associated with that, and then that can guide us when we look at other samples. So, the bigger scale we go to, the more confidence we'll have in those types of things.
And just to add to that as well, in our actual questionnaire as well, the one that the large one that's rolling out, we don't just ask whether you had a infection at the time of the blood draw. We will also ask when your most recent known infection was. So a patient may have had an infection 3 weeks ago. that they can then add in, and that would likely still be having an ongoing effect in their blood data. And then for those that could be months away, it may not be as present. So we do try to collect that.
Great. Another question is, has the decoded me results have any influence? Have they helped at all in your plans?
Yeah, they absolutely have. So when Decode.me released their genome analysis, their genomic data, which is really beautiful data, we had already received our RNA sequencing pilot data, and so it was absolutely fascinating. So we immediately started looking for the genes that they documented as being different, between patients or having impact on risk factor for patients, and we found several of those genes to be in our very preliminary pilot RNA sequencing data set, we found to be different already. So when we have our larger RNA data in January, absolutely we'll be querying every gene they reported. I thought that one of the things that was very interesting about their data was that, some of the genes that they found risk factors derived from were to do with kind of intracellular innate viral defense, right? So I thought that was really interesting.
All right, next question. Can you explain how or if people who do tests now will be included/rerun in future tests to add to the panels? Future tests you add to the panels.
Yeah, so that's also a great question. So I think that firstly, I'm going to answer that very clearly in a second, but firstly, one important thing is that every patient who has joined at any time will benefit from all future patients who join. So we've just created some really beautiful product data kind of visualizations from the 31 marker results for the people who joined batch 2. That's all going to go out to all our batch 1 patients and the comparisons between the wider patient cohort. As we grow, that will become accessible in real time through like a patient portal where you'll be able to see your results relating to all of the new patients. So that kind of continually means that you gain as we grow. In terms of retesting, that's a really good question. And what we're planning to do is one of the things about our company is that actually the more complex the data get, the simpler sample collection gets, because eventually it's just going to be a small amount of plasma or extracted RNA that feed into all of the different tests. So we have a biobank in Manchester in the United Kingdom, where everything is frozen. So that is where we keep the remaining sample from people who join. And so going forward, there will come a point where we're able to retain samples from every patient, and then we can retest on those samples.
But just to add to that, though, it would have to be a separate sort of order to retest. We wouldn't be able to continuously retest patients on our own. They would have to sort of like get the different products. Yeah.
Do the samples need special handling?
Yeah, they do. So this is something that we kind of put a lot of thought into. Basically, there's two kind of major paths that the samples need. So either if we're doing protein analysis, we separate the serum or plasma, and then that really needs to be frozen or cold from then. So we basically aim to have that arrive at our lab below, I think it's 10 degrees C, which I think is roughly 40 to 50 Fahrenheit. And then it goes in the deep freeze to -80, which is, I forget in Fahrenheit, I'm sorry. And so that we know retains, protects the kind of quality of the sample. The RNA has its own process, but this is something that my kind of background in clinical biotech development is really valuable, because we always had to protect drug products or different materials from deterioration. So I've been able to kind of bring that to how we handle sample quality.
And logistically for the patients, considering we know that most patients don't actually have the bandwidth to sort of go through the process of arranging the blood draw themselves, getting all the equipment. I remember I had to do, when I got GPCR antibody testing, I had to find a place to draw the blood for me, but then I also had to sort customs and FedEx and everything myself, and it took me about a month of emailing. So what we've done is we will ship the patient a blood collection kit, and then also we've set up a medical courier, so we do the shipping. We pick the sample up from the patient's house so that they don't have to deal with customs. And we wanted to make sure that that allows severe patients and very severe patients to join. So we've made it as simple as possible, basically.
Wonderful. I suspect your more tailored / sophisticated approach to trial groups could be applied to many autoimmune diseases where the patients can be quite heterogeneous, such as Sjögren's and lupus. Frequent drug trial failures, but there's almost always some percentage of positive responders. I'm surprised Big Pharma are not all over your approach. It seems like the way forward for a drug development in a wide range of immune and autoimmune diseases, and that's where the question mark is.
So, firstly, it really makes me happy that you can see how important this approach is. Like, I'm really glad that it comes across. We totally agree. We think it's insane. We think that the one size fits all trial design is crazy. I mean, one of the things from the industry is that, the kind of legacy systems, if you like, it's really need reform, but it's very complex to improve things. Um, but things are slowly changing. So we think, so obviously we think that long COVID and ME/CFS maybe need this type of approach more than anywhere else. And maybe that's why we exist, because there's no disease where it's more needed, because there's maybe no disease which is so complex and heterogeneous. But interestingly, in 2024 in the US, 30% of new drugs had a companion diagnostic as a part of the drug that was brought to market. So that means that it had some level of stratification diagnostic associated with a drug. So that would be like, you have cancer, you get a test, you find that you have a certain receptor on your cancer, you get a certain immunotherapy drug. And then I think most of those were in oncology. So that and oncology is where this has really been pioneered, interestingly. This approach is much more advanced in oncology than it is anywhere else. And I think over time that will spread out to other diseases. And obviously we hope to really accelerate that in long COVID and ME/CFS.
Great. Can you elaborate on what you're testing for related to aldosterone renin function? And the person commented, I have low aldosterone and don't know why.
Yeah, so for this one, so for the renin system, the RAS system, we test ANGI, AC, ANGII, AC2, and ANG1 to 7. So they're sort of the main components in the angiotensin aspect. And then what then typically creates aldosterone is ANGII will signal to create aldosterone. And then you actually have it's called the renin-aldosterone paradox, which exists in ME/CFS, which basically is we should be making more aldosterone and hence blood. But what the research has found is that we actually seem to be having really low aldosterone and possibly low blood volume. And this is where the paradox comes. There's certain pathways, like, for example, TGFB1. If TGFB1 is present at the same time as ANGII, it actually causes an almost 90% drop in aldosterone in mice. And then likewise, they can see up to a 50% drop in blood volume. So the angiotensin markers plus TGFB would be the immediate ones that I would say would relate to that system. We would then have to later on check blood volume, which is actually something we're looking to do in the future with a collaboration with a company. We will have to check whether stuff like TGFB1 correlates with stuff like that and aldosterone. But so yeah, the angiotensin markers and the TGFBs would be the main ones, I would say.
All right. Next question is, for patients who have done 31 marker tests, where do we see which of the 3 subtypes we've been categorized into?
Yeah, great question. So this will all be in your final reports, which will go out very soon. So in those reports, basically we have, I think it's 14, I forget how many, but I think it's 14 different clusterings. And so in the final section of the reports, you have each clustering, which looks exactly like the ones we shared today. Um, these are actually the clusterings that will be going out in the reports, and every patient has an ID for their has a patient ID, and so you basically will receive the clusters and your ID, and so then you can find yourself. And so right now you'll have that, and then you'll have the uh, kind of radar plot showing your kind of profile for this system. That's what will go out now, and then what we're working on in-house will be as we integrate the data together in the next 3, 6, 9 months, and develop our patient portal. Eventually, we'll want that you can come here and then see everything we know about your subgroup, right? So let's say you're in one of these subsets, then we'd like to be able to know everything that we know. So percentage of different symptoms in that subset, etc.
And something extremely important for patients to kind of understand with this as well is as we do more batches and as the sort of cohort size grows, even when we're in thousands of patients, these clusters will start to change slightly and more will appear. As we mentioned, we expect dozens of subgroups. Each patient who's even joined now will continuously be updated in relation to the expanding data. So even a year from now, when we're maybe reaching thousands of patients, you'll be able to log onto your patient dashboard and get a much deeper now insight into, there's 20 subgroups, I'm in this one, and we got way more insight into why this subgroup is the way it is. So it will continue to evolve indefinitely, essentially, until we majorly crack it.
All right, next question is a two-part question. The first part is question for Jack from a fellow long COVID sufferer, if he doesn't mind sharing. I was wondering how his muscle weakness manifested. I had long COVID for 2.5 years, after which I thought I may have recovered, but then started to experience relapses every 6 months or so. My latest relapse has brought muscle weakness with it, which is a new symptom for me. And the second part of the question is in regards to autoantibody testing. Did I understand correctly that this is on the future roadmap? And if so, do you have a rough timescale from introducing this?
Yeah, so regarding my muscle weakness, this was actually one of the worst symptoms I developed from immunoabsorption. I developed it about 3 weeks after immunoabsorption. It was almost instantaneous. I was just walking, and then suddenly I figured out I couldn't really walk much further. I tested my blood after, and I had a HHV6 reactivation, but this could also be a timing where antibodies return from immunoabsorption. So it's kind of like, could it be viral reactivation? Could it be autoimmunity returning in a new way? Hard to know, but yes. So for me, it was pretty instantaneous after immunoabsorption, a few weeks after immunoabsorption, and it's just basically never gone away, sadly.
And
With
then,
regards to the antibodies, I'll let Nick answer that.
yeah. So the second part, we're super excited about rolling out this autoantibody array, and we think that most likely it will be in the first few months of next year.
Wonderful. Another question was, do you have an interest in tick-borne diseases, or is that an otherwise nightmarish testing landscape?
No, we absolutely do. I think so we talk quite often about Lyme specific focus internally, but we also talk about focus on lots of different adjacent diseases. We think that, obviously our goal is to be kind of collecting the richest data in this area by far. And so naturally, these other complex, poorly understood diseases should benefit from that. And we very much want to work on them. I think important point to make is we actually although although we clearly have like a primary focus on long COVID and ME/CFS, we are interested in patients from all diseases. And so what we actually want is we want patients with chronic diseases generally to join our data sets. Right now we have I think we have only one MS patient, and we have a few who are post chronic Lyme. And over time, we want them to become, meaningful cohorts and to do research on them. So absolutely. We'll put more focus on it in the future, and I think, we'll kind of internally we'll focus more on good relation, making the partnerships with clinicians in the space and with, patient communities and community leaders in the space. But absolutely, we are super interested. I think at a high level, we basically want to work on all chronic neuroimmune diseases of any type.
And just interestingly, just for everyone's knowledge, there's actually a paper that was done by a really good researcher that used RNA sequencing of blood to try identify a blood diagnostic for Lyme disease, and they managed to separate control from Lyme patients. So as our Lyme cohort grows, it's very possible that we might find similar signals that allow us to potentially create a more definitive diagnostic for Lyme in time.
Do you remember how many samples that was? Or how many
I
patients?
think it was, off the top of my head, 80, but I could absolutely be wrong there.
Interesting.
All right. So if blood RNA sequencing allows you to see gene expression from the brain, would it potentially work to see expression from DRG and satellite glial cells in the peripheral nervous system?
Yes. Okay. Great question. I guess first thing is I can't be sure now. But it's very possible. So there's two layers to think about here. So one is, um, if we've got the sequencing data from the blood, the best situation is that we have specific genes that are only expressed in a certain tissue, right, or a certain cell type. And then if we know those specific genes and we can detect them in the blood, fantastic, we can answer that question. And so, satellite glial cells, super interesting. And, that's a little kind of afternoon of research for me to see, are there any glial cell, satellite glial cell derived specific genes that we shouldn't expect to see expressed by other tissues or cells. That can be quite a cloudy topic. Cell of origin and specificity in the body is generally like, I think people are overconfident about the where things have come from. It can be a lot more complex. So in one case, if we can find things that we know are cell specific, then great. We can say that we can see into that cell. The next thing is that interesting, right? So, okay, it might be cell specific, but does it give us interesting insight? I think this group of markers in this postpartum study was really interesting because in something like, in postpartum depression in the brain, there's probably such a complex and system-wide phenotype in the brain with depression of, different activity states and different types of neurons and microglial inflammatory states and blood perfusion and energy and all of these different things. So, from the thousands of genes expressed in the brain, it was, I guess quite probable that you would find brain specific genes that did relate to that disease state. Like I said, again, a very kind of pronounced disease state. The more niche you look, so the more specific you want to go, like looking into a single cell type, the more difficult that may become. Caveat to that, though, is that tissue specific can be easier, right? So if we're looking at, generally gut derived or muscle derived gene expressions, then the chances that we have, both specific genes and ones that carry interesting information is higher. There's a third caveat that I'd add. I mean, obviously you can tell the short answer is we don't know yet. But the third caveat that I'd add, which I think is also really interesting, is that there's a really interesting separation for us, which we are very aware of in our work, which is um, if we crack a mechanism, if we identify, let's say some genes that have a mechanistic contribution to a disease state, fantastic, that's brilliant. That could become something we can learn from and tr design treatments around or diagnostics. It's obviously the best thing, but we don't have to make mechanistic findings to detect signals that tell you about different systems as well. So, for example, if you're interested in pathological satellite microglial states, that even if we can't see into them directly and get direct information from them, there is probably an entire like storm of different signals that go with those pathogenic states, either from the cells themselves or from the surrounding tissue or from up or downstream systems. So when we do our research, we can kind of be completely agnostic and say, okay, what signals do we find overall? And then point two, do those actually relate specifically to pathogenic mechanisms? And if the answer to point one is that you find signals, you've already made progress because the signals can become diagnostic, even if they don't relate to mechanism. But of course, if they do relate to mechanism, then that's even more exciting.
Thank you. All right, so next question. Every time I click down on a question, another one pops up, so the queue is not going down at all. How do you view the stability of the 31 biomarker and RNA results? Is there a risk that some of them can change significantly across different times of the day or different days? Also, can you comment on the stability of patients pertaining to a given subgroup? Is it possible that a patient could move between groups over time?
Great question. So firstly, we know that there's a lot of variation inside the body throughout the day. I think that the way doing data science in human biology, you are always fighting against a huge amount of variation in the state of the body at the time of getting a sample or in various different things that can happen to a sample. You do everything to combat those things, but the best way to combat it is to look for is to design good ways to validate what you think you're looking at, right? So, we wouldn't just take so for example, we can cluster the 31 marker patients and get these interesting subgroups. But we don't just do that. We look at those subgroups and we say, can we identify disease features that map to those subgroups or that correlate with them? And the more you can do that, the more confidence you can take in the findings. Now, for an individual patient, the way you're asking this question, if you have had the 31 marker test, you can't be sure that, we don't have data on the stability of every marker across the day. I've worked on circadian rhythms in my past, and, I know there's a huge amount of variation, but um, but we don't just look at one marker from each system, right? So within the 31 marker panel, there's almost always at least one complementary marker, if not more. And then things kind of relate to each other in networks. So although, variation in one marker should be offset by that to some extent, then relating it to disease profile and disease states will give us more confidence. But but bridging to the second part of your question about whether a patient could move from one subgroup to another, patients will change a lot throughout the day. And we are really proud to be collaborating with, I think, the best ME/CFS researcher in the world, Alain Moreau, Professor Alain Moreau from Montreal. And his work has looked at the temporal and state specific signatures of PEM, for example, right? And over time, that could be a really important thing. We will be able, and right now we don't know the answers to those questions, but by looking at associations to symptoms and by looking at metadata, we'll hopefully be able to answer those things and be able to give that back to patients as valuable information. For example, we do have the data on the time of collection for each of these samples, etc. So, ultimately, if we have thousands of samples, we'll be able to separate samples by collection time. And, I think we, as a company, we really believe in following the data. And so, if we find that, some marker is extremely affected by collection time, we will make that transparent immediately because we think it's all super interesting in finding the answers that we need.
Next question. Will the autoantibody array include muscarinic acetylcholine receptors, adrenergic receptor autoantibodies? Will it be able to identify functional autoantibodies?
Great question. So the first answer is yes, it will include every different type of receptor. So that 21,000 array, and just to reiterate one more time, it contains within it uh, it contains within it 21,000 human proteins which have been bound to the wells of this microarray. And then your serum is put on top. And if you have autoantibodies against any of those human protein targets, your autoantibodies will bind and then they, wash and then add a fluorescent tag which will tag onto the tail of the antibody of interest. So we can tag IgG or IgM, etc. And so that means that we will see, do you have autoantibodies that bind to a specific human protein target? We can also see intensity, right? We can see how many antibodies bind. So you might have a stronger antibody profile against certain receptors. Also because of the method of preparation, there will be some proxy of strength of binding, right? Because the signal that ends up in the final analysis moment will also be a function of how strongly the antibodies bind, right? But we cannot infer from that directly whether they're functional or not, right? We know we can tell if they bind and the maybe strength and intensity they bind with. Knowing if they're functional has to be follow-up work, but we think we'll be able to make really cool progress there because when we layer this together with people's patient profiles, let's say imagine from thousands of patients we see, 500 who have antibodies against GPCR receptors like the ones you mentioned. We then maybe will see that they have certain specific symptoms more than people who don't. That can add confidence in whether those antibodies or whether those autoantibodies are functional, if you like. Equally, when we do from the RNA sequencing, we will integrate that autoantibody array data with the RNA sequencing data, and that will be extremely powerful because if you have, let's say you're positive for a muscarinic receptor autoantibody, and you also, so we see that in the autoantibody array. Then in your RNA sequencing, there should be associated signals, right? We should see differences in gene expression to do with up or downstream things or the receptor itself. So on an individual basis, it will take us some time, but that's the type of data science that we'll be doing in, let's say, in the second half of next year and beyond. And that should be able to answer questions. And I think one point I'll add here is we are engineering a data set, if you like, to try to answer these questions. And if you engineer the data set well enough, you can do experiments in the data itself, which is how bioinformatics works. So, the question you asked, that could be a simple bioinformatics experiment where you say, okay, let's look at everybody who has a muscarinic autoantibody. Do we see associated gene expression changes? Do we see associated symptoms? And if the answer is yes, then that might tell you if it's functional, and maybe we'll be able to separate patients into functional groups. That's all possible. We'll need some time and hard work to get there, but it's a really good question.
All right. If people with ME and long COVID have been able to apply their Amatica results to hone in on potential treatment pathways, could you share any anonymized specifics?
Sorry, could you repeat the question?
if people with ME/CFS and long COVID have been able to apply their Amatica results to hone in on treatment and treatment pathways. Could you share any anonymized specifics?
Yeah. It's a great question. So we actually do have some internal discussions about this. Could we create an anonymous a way to get that data out there quickly? So we have a few things to think about. So firstly, the first thing is that there's, unfortunately or fortunately, strong legal framework around how, clinical data is allowed to be put out there, right? So we have to think about that. We have to take care that we are really confident in what we put out there as well. So, as someone who's worked in academics and industry for a long time, I spend a lot of time hating the legacy clinical development systems. One caveat to that, though, is that, clinical validation done right is about validation. It's about really ensuring that you have confidence that the treatment signal you have is real. So, firstly, anything that we put out there, we have to be sure that we're putting out information that genuinely is going to help people and not harm people. That's one step, and there's many ways we can achieve that. Um, but I think the way we will achieve it fastest is so we actually have a pretty developed plan for this. So and this is a good full circle to the topic for today. This is why clinicians are so important because the position that we can take is we can collect all of this data and then with the right uh, processes in place and accreditations, which we're taking care of everything, we can give that information to clinicians, right? So we can say to clinicians, this patient we believe, and again, this is not diagnostic and it's not a recommendation for a treatment, but we believe that this patient has a higher probability of responding to this drug than that drug. That precedent has been established in oncology, and we study very closely how that was established because the company that pioneered that, Tempus Labs, penetrated 50% of US clinics and was, onboarding hundreds of thousands of patients a year to do exactly that, to accelerate the process of getting that treatment response data back into the hands of clinicians to be used in patients without going through years of clinical development and doing formal clinical trials. So we are engineering the same system. And the point of contact I'm most confident to say that we'll be able to do is going to doctors. And so we hope to kind of expand the network of doctors we work with. We also don't want to leave anyone behind. And so, I think that what we will also be doing is basically, like, for example, today, presenting what we have in a fair and grounded way. Right now our signals are very early, but I think we will do regular seminars and conversations where we present what we have. So not making any claims about treatment impact and grounding everything in the limitations of our data and being very clear about what we don't have confidence in. But we very much want to solve that problem to get the utility of our data into the hands of the patients who need it so much.
Thank you. We are out of time, and I was told that the questions that remain in the queue will be saved, so there's no need for emailing them. Thank you, everyone, but special thank you to the Amatica team for this presentation today and for all the wonderful work that you guys are doing. And to follow them, make sure that you go to their website, their X account at Amatica Health, and their individual accounts on X as well to keep on informed about everything that they are doing. Thank you so much, guys. Bye, everyone.
Thank you so much.